So our second speaker in this section is Dr. Ann-Katrin Piper from the University of Wollongong in Australia. Unfortunately, she couldn't join us in person, but she is online, so she'll be able to answer some questions later. Ann-Katrin is a postdoctoral cancer researcher at the University of Wollongong, and she completed her bachelor degree in Hamburg and master's in Oldenburg. And she did her PhD at the University of Australia. So Ann-Katrin is going to tell us about her research about the potential role of CLCA1-ANO1 Axis in supporting survival of gastric cancer circulating tumour cells.
Hello everyone, my name is Ann-Katrin Piper and I'm a cancer researcher from Wollongong, Australia, down here about an hour south of Sydney and about 16,400 kilometres away from where you are today. First of all, I would really like to thank Tom for the kind invitation to contribute to your fabulous meeting today. And I was really hoping to join in person, but it didn't work out this time. Hopefully next time. The title of my talk today is the potential role of CLCA1 and ANO1 in supporting the survival of gastric cancer circulating tumour cells. First of all, I would like to give you a bit of background about the metastatic cascade. That is the process during which the cells detach themselves from the primary tumour, travel through the tissue, and squeeze themselves into the blood vessels through the endothelial wall of the blood vessels. This process is called intravasation. Now they're in the bloodstream, and here they are called circulating tumour cells, and this is when they're at their most fragile. And only 0.01% of circulating tumour cells actually survive the journey through the bloodstream. And why that is, I will explain to you on the next slide. But this 0.01% of cells are actually the responsible ones for killing up to 90% of cancer patients, because they are the ones that end up seeding metastases. To do so, they then have to travel through the bloodstream and squeeze back out of the blood vessel wall, a process called extravasation, and then seed new metastases at distant sites. So why do all these circulating tumour cells die and only a very tiny fraction makes it through the bloodstream? Well, that is partially due to the huge amount of stresses that they are exposed to while travelling through the bloodstream. These include shear stress, immune cell attack, collision with other blood cells, and resistant to anoicus, death due to loss of attachment. But all CTCs coming from solid malignancies from all different types of tumours have to travel through the bloodstream or the lymphatic system, but today I'm going to focus on the bloodstream. So somehow they must develop a coping mechanism that allows them, this very tiny fraction of 0.01% of cells, to survive the bloodstream. Because only here they are exposed to the same microenvironment, and maybe all CTCs have a coping mechanism in common. And what that might be, that's the focus of my project. And whether or not ion channels could be involved, this is the focus of my talk today. Because as I said before, during the metastatic cascade, there's a lot of pressure on the circulating tumour cells, on the tumour cells that are seeding the metastases, squeezing through the blood vessel in and out, adjusting to mechanical stresses. All of these things are ion channels are involved in. So therefore, it is a plausible explanation that ion channels may also aid the survival mechanisms during the bloodstream journey. As you all know, ion channels are playing key roles in all physiological processes in our bodies, but they've also been known to play key factors, or are key factors, in cancer biology. And here they have shown to contribute to all hallmarks of cancer. And these hallmarks include things like chemoresistance, cancer cell proliferation, migration, invasion, tumour vascularization, as well as metastasis. So therefore, I believe it's also very possible that ion channels play a major role in the survival of circulating tumour cells. To study circulating tumour cell specific adaptations, we have a fabulous set of tools in our lab. My supervisor Daniel Brunks, during his PhD, were able to derive a circulating tumour cell line from the blood of a gastric adenocarcinoma patient called UWG02 CTC. And at the same time, he was able to derive a second cell line from the same patient, taken as a simultaneous biopsy, derived from the ascites infiltrate. So this is when the tumor from the stomach directly breaks into the peritoneal space and resides there. And he was able to take this ascites fluid and established a second cell line. So this cell line has never traveled through the blood and is therefore a fantastic control for our CTC-specific adaptations. And another cell line was established at the same time from a gastric neuroendocrine cancer, which is a very rare cancer, but we've studied the cell line alongside these cell lines as a control for circulating tumor cells. We performed several different analyses like bulk RNA sequencing, lipidomics, and proteomics, and found a couple of very interesting differences between the ascites-derived cell line and the CTC cell line. But for the interest of this meeting, I would like to focus on the ion channel bit with a particular focus on ANO1 and CLCA1. Before I dive into the results further, I would like to give you a little bit of cancer-specific background on ANO1 and CLCA1. So ANO1 is anoctamin 1, or also called TMEM16A, and it's a calcium-activated chloride channel, which is usually highly expressed in the gastrointestinal tract, where it's involved in mucus secretion, and is often overexpressed in cancer. ANO1 is particularly involved in a process called regulatory volume decrease, which I think might be quite interesting for this particular setting of circulating tumor cells. As you can see here, ANO1 is known to play various roles in cancer, where it's involved in signaling, metastasis, as well as its ion channel and role, as well as interacting with EGFR, the growth factor receptor, for instance. And it has been discussed in the literature as a potential new target for cancer treatment. CLCA1, or the calcium-activated chloride channel regulator 1, is known to stabilize ANO1 at the cell surface, and where it's then increasing its chloride currents. We first started to be interested in CLCA1 because it was one of the only differentially expressed genes on RNA sequencing between the circulating tumor cell line and the ascites-derived cell line. And when we looked into the literature, we saw that CLCA1 was implicated in different cancer types, but its role specifically was a little bit controversial, as there was one review stating whether or not it should be a tumor suppressor or an oncogene. And I think that's a quite interesting aspect, considering that we're only seeing CLCA1 in the CTCs and not in the ascites from the same patient. We then looked at calcium-activated chloride channels in particular, and could see that ANO1 was expressed on similar levels between the circulating tumor cells and the ascites-derived cell lines, and CLCA1 was slightly different between the two on RNA level. And we were quite excited then to find, when we performed Western blot analyses, that the slight difference in RNA expression resulted in a pretty much black and white expression on protein level for CLCA1. But for ANO1, we saw similar protein level expression between the UWGO2 CTC and the UWGO2 ascites, and the neuroendocrine cell line didn't express either of them on protein level. We then wanted to go ahead and investigate what that could mean and what the functional consequences of this may be, and luckily we have access to a SynchroPatch 384 PE here in Wollongong, and in collaboration with David Adams and Rocio Fino Urdaneta, we were able to perform this particular assay developed by Nanian. So using our cell lines, we recorded whole cell currents in response to a RAM protocol simulation between -100 millivolts and 100 millivolts, using the SynchroPatch PE 380 PE to assess calcium-activated chloride currents. When internally perfused with a solution containing nominally 0 calcium, these cells revealed minimal chloride currents, as seen here in that grey line. However, when we added 4 micromolar of calcium to the internal perfusion solution, we could see large outward chloride currents, as seen here in green. And importantly, when we added 250 micromolar niflamic acid, these could be almost entirely blocked. And when we did the same thing to the ascites-derived cell line, we could see that the maximal chloride conductances were less than in the CTC line. And as expected, the neuroendocrine-derived cell line that didn't express any ANO1 or CLCA1 pretty much showed no calcium-activated chloride occurrences. We were excited to see that when we performed statistical analysis that the results were highly significant, showing that the CTC-derived cell line exhibited significantly higher calcium-activated chloride current densities compared to the ascites-derived cell line and the neuroendocrine CTC cell line. We repeated this experiment a couple times because it was kind of too good to be true, and we saw the same result again and again. And that led to us taking a stroll to the Unibar. This experiment was performed with Rocio and Tom, who was there to look after our instrument during that time and assisted Rocio on performing this experiment. And we were so excited that we had a beer to cheers to this experiment. And what does this actually mean now? Well, we think that given only the CTCs express CLCA1 on protein level, that is stabilizing ANO1 at the surface, as the literature suggests, or somehow increases its chloride conductance, and that may aid the cells adapting while traveling through the bloodstream. So in summary, today I've shown you that CTCs utilize unique molecular adaptations to overcome challenges during the metastatic cascade, particularly during the time of travel through the blood. And our CTC cell line express high CLCA1 protein, which resulted in the increased calcium-activated chloride occurrences. And we believe that this mechanism is likely to contribute to CTC survival through the mechanism called regulatory volume decrease. And we're currently in the process of investigating this further. And I think that we can go as far as saying that CLCA1 or ANO1 may present promising targets to stop CTCs, and that this might be an interesting field of investigation for the future. And with this, I would like to thank all my team here in Wollongong, and in particularly Rocio and David Adams. And also I would like to thank Tom, who helped us tremendously setting up this essay. And you all for listening, and I'm happy to take any questions. Thank you very much.
Thank you very much, Ann-Katrin. I believe that you are online, so we have some questions from the audience.
Hello.
Oh, yes, there you are. Maybe I can start with one. Ann-Katrin, is there any evidence that the CLCA1 is like mechanoactivated? You would think it may be, with it squeezing through, the cell squeezing through, would they have any mechanoactivation?
I'm not too sure, to be honest, about CLCA1 itself being mechanically activated, but I think the mechanically gated channels definitely would play a role, or it's already known that they do play a role. But it's just interesting too that CLCA1 itself also has a role in being a metalloproteinase, so that's really important in the metastatic cascade for digesting the extracellular matrix of the target organs for the metastases. And I think that that's partially its role also on the circulating tumor cells, maybe to groom the target organ of the metastasis after its survival through the bloodstream.
Any other questions?
Good day. I've got a question. I know a lot of people are big fans of looking at gene expression, protein expression, and those don't always go hand in hand. You can get opposite changes. But I'd also be interested in modulation. So, for example, does the calcium dependence of the ANO1 change in disease? Have you looked at the calcium dependence in your experiments?
That's a very interesting question, and I don't believe we have, but I must admit I'm not an expert in this, and that's definitely Rocio who would be much better at answering this sort of question. But we did use, we could potentially look at that, I believe, because we used a 4 micromolar calcium internally perfused solution. So I believe we could probably adjust that, but I'm not sure how easy it is to tweak this kind of assay, because I think it was quite exciting that we actually did get it to work the way we did. But I'm sure there would be other ways in potentially figuring that out.
Hi there, another sort of question related to potential ion channel function. Have you tried, pharmacologically inhibiting the target, or thought about whether blocking it might be enough, or whether you actually have to down-regulate the protein and just take out some interaction that it's making within the cell?
Yeah, that's a very good question. We have actually blocked, so with meflomic acid, which is a broad inhibitor of all calcium activated chloride channels, but we also used NE9 to specifically block ANO1, and we saw some interesting differences that I didn't really present today because they're not quite ready yet to be fully presented, but I can tell you. So these cell lines have a peculiarity about them because they grow in a half attached, half suspended phenotype. And Rocio has taken the cells in different experiments where she's only looking at the suspension cells versus only the attached cells. And she could see that the suspension cells have a higher chloride currents than the attached cells. And we're going to now go ahead and do flow cytometry analysis, and we want to see whether or not the increased expression, or whether or not the CLCA1 is enough to increase the chloride currents to cause shrinkage of the cell through the process of regulatory volume decrease. I'm not sure if I answered your question, but I just wanted to say. Sorry. I kind of went off here. Sorry.
No worries, Anne. We've got one question here online. It says there's debate at the moment whether a protein degrader is needed for anti-cancer activity of ANO1 versus pore blocker. What are your thoughts on that?
That's an excellent question again. As I said before, I'm not an expert in this, and it sort of came about as a exploratory quest of what is different between these two cell lines. And NR1 itself wasn't actually that different. As I said, the expression, protein expression, doesn't seem to be so different. So I'm not sure, to be honest.
Thank you so much, Ann-Katrin. That was really amazing. I particularly enjoyed the picture of you enjoying a beer with Tom. Unfortunately, you can't enjoy a beer with us tonight, but I guess you can go to bed.
I would have loved to, yeah. Thank you very much for having me. It was a pleasure.